Ladies and gentlemen, now we are starting the session 3. Now let's welcome the chair of this session, Professor Fang Ren Li. He is a distinguished professor of Institute of Molecular Medicine, College of Medicine, National Taiwan University. Let's welcome Dr. Li.
Welcome back to the Section 3 therapy. It's my pleasure to chair this section. And today, this therapy section have 5 speakers, and 4 will be in person, 1 from the virtual. And our section will cover from the vaccine and diagnosis and therapeutic antibody special for IgA, and drug development, neutralizing antibody, and to the drug representation for the COVID-19. Okay, now let me introduce first speaker in this section is Dr. Jason Wang, and we are very pleased he flew away from the United States back to Taiwan and give out this presentation. Dr. Wang currently is a director of Center for Policy, Outcomes, and Prevention, and also is associate professor of Pediatric Medicine and Health Research and Policy at Stanford University. And let's welcome Dr. Wang.
Can you hear me now? Yes. Okay. I want to thank the organizers for inviting me back to Taiwan. And so the title of my talk is Navigating COVID-19 Vaccine Procurement Under Uncertainty. And I'll give you examples for high, middle, and low-income countries. So earlier this year, we shared Taiwan's success story with the world. And I'm pleased to say that currently this article has received more than 1 million views. And so Taiwan's success story has been shared all around the world. So let's get back to the issue of vaccines. As you know, the situation currently is that every month that this pandemic continues, $375 billion is lost from the global economy, not to mention the already over 1 million deaths all around the globe. And so now in terms of vaccines, the probability of success for vaccine in early stages of development is less than 20% before stage 2, and vaccines overall have a success rate of 33%. So it's a very low percentage. And as earlier speakers have mentioned, nobody is safe until everybody is safe. Really, vaccines ought to be an important part of the solution to reopen the world. Vaccines must be at least 50% effective for it to get approval from the FDA. That means it needs to protect on average at least half the people who take it. In naive population, 70% of the population must be vaccinated to achieve herd immunity. And experts have said that 2 doses of vaccine may be necessary. And so, for example, in the United States, 462 million doses will be needed to achieve herd immunity, and 660 million doses for the entire US population to each get 2 doses. So that's a Herculean task. The value of the vaccine really is that it maximizes near-term impact of the COVID-19. So right now we have a pandemic, and if we could get people vaccines, maybe it will narrow down to an epidemic and maybe even to localize in certain areas of the world. In the midterm, we hope, in fact, that COVID will become an endemic, and even though it continues to circulate, it will require routine vaccinations. And in the long term, development of the vaccine, as a previous speaker had mentioned, could prevent sort of the next epidemic or pandemic if the target is correct or if the therapeutic area for therapies are correct. Now, I've told you the situation, but there are complications to the situation. So no pharmaceutical company could make 5 billion doses in the next year. The cost and distribution system, including cold chain requirements and the delivery of widespread coverage for all the potential constricting points, is going to be what limits the delivery of vaccine to individuals and communities. And that is a quote from Dr. Anthony Fauci. Different vaccines have different logistical needs, include the dosing schedules, the site of administration, and the cold chain requirements. And so for DNA-based vaccines, which are traditionally shelf-stable in normal temperatures, so those are easier to sort of ship. But for RNA-based protein subunits and viral vectors require cold chains or cryo shipping. So you need to have refrigeration, make sure during shipment it doesn't degrade. And these are just to get the vaccines there. And once you get the vaccine there, people have to be willing to take it. So currently, only about 30 to 50% of the individuals in the United States will be willing to be immunized. And if we look at the subpopulations, only about 25% of African Americans and 37% of Hispanics in the United States are willing to take the vaccine. Okay. In order to meet the production demand, industry needs to start preparing to manufacture in parallel with research. So this has never really happened before, long before clinical trial results are available. And to make this happen, there needs to be some relaxation in the regulations and with a post-vaccine monitoring. So the issues to be addressed at hand are multiple. So for one, you need to find a vaccine that works. And it's expected out of the 250 or so in the pipeline, about 7 to 9 will be available in the next year. So the size of the pipeline is vast. Okay, a lot of investment has gone through this. The speed has also been unprecedented. So it took 4 years to develop the mumps vaccine, which was previously the fastest. Usually it takes 10 years. Okay. The first candidate for a vaccine for COVID-19, SARS-CoV-2, was 42 days after the genetic sequencing that was proposed. However, even though the pipeline is vast, there's a first mover advantage. So imagine the first vaccine has been shown to be effective. A lot of investment will go into that company. So it's not equal footing. It's not like you start with 250 and then you end up with 250. You're going to probably end up with 10. The mutation rates, which has been discussed earlier today, if it's moderate and there's relatively little effect on antigenicity, and then the vaccine is going to work well. And many people have used the spike protein as the target. In this round, the vaccine development, there's been lots of unproven technologies. So for messenger RNA and DNA, these are mostly unproven, they are fast, but we don't really know the long-term safety or the long-term effectiveness in terms of lasting immunity for these vaccines. For the more proven platforms such as protein subunits and viral vectors, we have some earlier data on those. Vaccine development also depends on sustained attack rate. So they say if there are no infections currently in a country, it's hard to develop the vaccine. So you actually need to do the clinical trials in areas where there's still infection that's going up. And then the indication of protection include neutralizing antibodies or protection against actual disease. So for any regulatory body, and I'm a policy analyst, so I think about these things, you need to consider the risk and benefit ratio. For high-risk populations, I think it makes sense. For low-risk populations, for example, for children who have relatively mild symptoms in general and fewer cases of serious disease, we need to be more careful. The production at massive scale is an issue, as I mentioned, and definitely the widespread distribution. There are many places in the world where it's hard to get to, but they do harbor the disease. So we need to develop a system of delivery and a system of tracking, because there could be more than one vaccines that's available. So let's look at this. This data was pulled on September 25th from the New York Times, and they have a very good tracker. I encourage you to use it. So right now, there are 11 vaccines in phase 3 clinical trials. So for the audience here, phase 1 usually is a safety study, small numbers. Phase 2 is an expanded study that usually includes children and the elderly to see how they react. Usually is in hundreds, and phase 3 usually is in thousands, if not tens of thousands. And typically in this pandemic, people are using 30,000 or 40,000 for phase 3 clinical trials. There are 5 vaccines that are currently approved for limited use in selected countries such as Russia and China. So I want to walk you through the 11 vaccines that are currently in phase 3 trial. And I want you to decide what to do for Taiwan, which should be obvious at the end. Okay. So the first one is Moderna. Everybody knows this one. It's a messenger RNA vaccine. It's in phase 3 trials. The US government has invested close to $1 billion for R&D, $1.5 billion for its supply. The advance purchase for this vaccine, the US had acquired $1.5 billion, used $1.5 billion for 100 million doses with the option to buy another 400 million doses. Okay, and Canada had also made advance purchase. So BioNTech, a German company working with Pfizer, a US company, and Fosun Pharma in China, the US had invested $1.95 billion and had acquired 100 million doses by December with an option for another 500 million doses. Okay? And CanSino Bio, now this is developed in China as an adenovirus vector, which is different from the previous mRNA vaccine. And so there's no information. We don't know much about it, except it's approved for limited use in soldiers. It's just beginning phase 3 clinical trial, so it's approved after 2 phases, phase 2 clinical trial. The next one is by Russia. It's called Sputnik, like their previous spaceship. Okay, this is the most popular one, a lot in the press. It's also an adenovirus vaccine. It's also in phase 3 clinical trial, but it's been approved for the use. There's no information that we could find. But Russia agrees to supply to Brazil, Mexico, and India. Okay. For Johnson & Johnson, the US government invested about $500 million for BARDA and another $1 billion for manufacturing and delivery. And with the advanced purchase of 100 million doses with an option of 200 million doses. Do you see a pattern here? The US is going to get all the vaccines. Okay? And AstraZeneca, US, invested $1.2 billion. It's a chimpanzee adenovirus. Okay. And Europe is going to buy 400 million, US 300 million, UK 100 million. And a lot of it is going to be made in the Indian Serum Institute, the largest manufacturer of vaccines in the world. They do manufacturing. They license and they do manufacturing. Okay. I want you to pay close attention to this Indian Serum Institute. And then Novavax, again, is now a different technology. We were doing viruses, now we're doing protein-based. CEPI invested $384 million, US government $1.6 million. This is based in Maryland in the United States. US is going to buy 100 million doses, and the Indian Serum Institute is going to make them. The next one, Sinovac, the China vaccine, inactivated. It's approved for limited use in China. It began phase 3 clinical trials, but it's already approved. Very little information. Indonesia is going to get 40 million doses. Okay, next slide. Now these are the Chinese vaccines. The Wuhan Institute is using inactivated virus as a vaccine, and it's in phase 3 clinical trials. UAE gave emergency approval. So UAE is going to use it right now. No information about the data on clinical trials. Beijing Institute for Biological Products, Sinopharm, also inactivated. UAE also gave emergency approval for healthcare workers. The Murdoch Children's Research Institute repurposed the BCG vaccine that's thought to have some cross-antigenicity and protective effect, and they're doing phase 3 clinical trials. No information. Okay. Now, a lot of information about who's going to do advanced purchase, but let me tell you about COVAX, because Taiwan signed up for COVAX, correct? Okay. So COVAX is part of the ACT accelerator for coronavirus toolkit. COVAX is the vaccine part, the vaccine pillar of ACT accelerator. The COVAX involves Gavi, the Vaccine Alliance, that they do mostly procurement and allocation, and CEPI, the Coalition for Epidemic Preparedness Innovation. They do mostly R&D and coordination, and the WHO. The goal is to have portfolio diversification and pooling of financial and scientific resources, and to bring it to scale. So the idea is that most countries will not have the resources to bet on an individual vaccines like the United States, and then they need an agency to do that for them, to hedge the risk of backing unsuccessful candidates. I told you it's 33% success rate, and if you're before phase 2 is only 20% success rate. The government with limited or no ability to finance their own or to do bilateral procurement agreements could have access to the vaccine. The goal of COVAX is actually by the end of 2021, next year, to deliver 2 billion doses of safe, effective vaccines and to deliver them equitably based on population to all participating countries, initially prioritizing healthcare workers and expanding to 20% of the population. I read in the UDN news, 联合报, yesterday, that Taiwan has to pay close to 100 million for 4.6 million doses, about 20% of the population. So the payment is due on October 9th, I believe. That's what they say in the news. Okay? The COVAX facility is a key part of COVAX pillar. It involves 165 countries, 75 from more well-to-do countries that are going to be paying, 90 from lower income countries that are going to be supported by this global initiative. The advance purchase agreement to the vaccine candidate has to meet the WHO's technical threshold for an effective vaccine. And they recommend prioritizing populations such as healthcare system workers. That's 1% of the global population, adults over 65 years old. It's about 8% of the population, and other high-risk individuals with underlying conditions. Okay. Now let's look at COVAX commitments. Okay. So as of right now, Gavi had launched the Advanced Market Committee. And the countries that are going to be participating are these countries: Canada, the ones that you recognize as vaccine-producing countries, Switzerland, United Kingdom. Who's missing? Everybody is missing. United States is not part of COVAX. China is not part of COVAX. Germany is not part of COVAX. Russia is not part of COVAX. These are the countries that make vaccines. They are not part of COVAX. Okay? So the money raised, they need about 15 billion, I read earlier today. They get, today it was updated to 3 billion. So they got 3 billion out of 15 billion. Okay? The committed doses, they only have 300 million doses. Okay? I show you the chart from what US is doing before. This is what US is doing. US currently has acquired 850 million doses with an option to buy another 1,100 million doses. Okay? Assuming you bet this 33% success rate, US is going to get 280 million doses. That's going to cover 85% of the US population. You only need 230 million doses to reach herd immunity. Assuming that you need 2 doses each, the option to buy 850 plus 1,100 million is 1950 million. They still going to have enough. They're going to have 643 million doses, almost enough to cover the entire US population twice. Okay, this is where Taiwan is. Taiwan has 3 vaccine companies: Adimmune, 国光生技, United Biomedical, 联雅生技, Manage Vaccine, 高端疫苗. The government has invested $6.8 million US dollars into 国光生技, 15 million into 联雅生技, and there's no information on advance purchase that we could find. Okay. Now, there are factors that would determine if the vaccine is valuable, and one of them is that it has high levels of protection, that it has extended duration of immunity, has simple logistics for distribution, and you don't need a lot of complicated shelving or shipment or thermostable, and has a convenient administration. And it has hopefully that it will have if therapeutics to come along, that's very effective. You don't need the vaccine that much. But if there's no good breakthrough therapeutics, then you really need the vaccine. Okay. And then a vaccine is very valuable if there's constantly moderate attack rate, so that it spreads really fast. Okay. Now, given that COVAX, I just told you, have very few vaccines committed and very little funding. Okay. And US has a lot of vaccines committed, a lot of funding. What can Taiwan do? I told you that this India Serum Institute, remember that one? They make them. They help to make them. Okay? So you have to map the production capacity. And usually where people get stuck is the fill-finish capacity. That's when you have the vaccine, but you need to package it in the vials, and you need to make sure it's sterile. And that's usually the bottleneck of this entire process, that people already have the vaccine, they can't package it in a sterile environment. So we got to get on that one, okay? Coordinate vaccine manufacturing capacity and supply chain manufacturing capacity for vaccine supplies. Usually people run out of wipes, vials, syringes, needles, stoppers, adjuvant. Now, if Taiwan could help with that, we could negotiate, okay? And you need to basically figure out how to cover the cost of the vaccines. And I think because of national health insurance, I think Taiwan will be okay with that. For the United States, we actually have to work with the insurance companies and to make sure that the insurance companies do in fact have cover the cost. Or otherwise, the government would need to. And then where are you going to give the vaccines? You need to have vaccination clinics. So if you want to deliver the vaccines in large volume, we've never done this before. That where everybody would need a vaccine. And you need to establish a tracking system and a vaccination campaign, because I've just told you that half the people in the United States do not want the vaccine. 75% of African Americans do not want the vaccine. 60% of Hispanics do not want the vaccine. Okay? So you need to have a campaign to trust is part of vaccines. If I don't know what you're giving me, why am I taking it? I'm healthy right now. Okay? So we're going to move into strategy now, how to do strategy with vaccines. Okay? So when you're managing uncertainties, you need to identify the nature and extent of residual uncertainty. You need to choose a strategic posture. You need to build a portfolio of actions and then actively manage the strategy. Okay, so if you're the government, you have to do this. This is your job. Okay. So if you have a clear pathway that you know vaccine A is going to work, you just go bid on vaccine A. But right now, it's not the case. You have 11 vaccines, each with a 33% chance of success. You don't know which one to bet on. So you have all these alternative pathways. Okay? And different vaccines may play different roles over time because the vaccines could have different characteristics. One could allow you to have immediate protection, the other one could last longer. You need both, right? So it depends on the characteristic of and they may be complements. So it's pretty clear that you need some sort of a vaccine to reopen society, okay? You can't just open. Or you need major advancements in therapeutics. So what could Taiwan do? One is you have to figure out the science of reopening, okay? Because right now, as far as I know, you're probably not gonna get any vaccines by the end of next year, given what COVID has come up with. Okay, because there's so few committed. And so unless Taiwan is gonna close for another 4.5 years, you need to figure out what is the safest way to reopen. Not right now. I'm not saying right now. Right now, it's not safe. Gradually, you have to have travel bubbles, travel corridors, slowly reopen with other safe societies. And you could do that with serial testing and enhanced contact tracing that Taiwan does very well. The other one, you have to build flexibility and fungibility in the manufacturing process, as I've talked about. You have to be able to convert the facilities into making other people successful vaccines if your own is not successful. And if right now, Taiwan's 3 manufacturers is in phase 1, so probably by the middle of next year, you'll probably be in phase 2 or 3. Phase 3, you have to do it outside because there are no cases here. You cannot prove efficacy. So you have to work with other people with cases outside. That needs some time to set up. So that means in the meantime, you have to figure something out. And maybe the fill finish, the manufacturing process that Taiwan does so well, you could do. Okay. And then COVAX. Taiwan has already committed to COVAX. So Taiwan is going to get some vaccines through COVAX, but I don't know when. Because, it depends on how long COVAX get the vaccine. And so Taiwan might need bilateral agreements. For example, with the United States. Okay. Other countries might have it with China. Other countries might have it with Russia. These are the vaccine producing countries that are not part of COVAX. So with COVAX, you get 2.3 million, 10%, and if you buy 20%, that's 4.6 million. And be prepared to contribute to the manufacturing so as a negotiation power. You might need bilateral agreements or multilateral agreements with the US or UK. And the big bets are the domestic vaccines, which you will need because viral infections are going to happen over and over again in Taiwan because of its close proximity to China. So you have to get prepared. But it takes time to invest in vaccine development. So I'm going to stop right here. I probably went over, but probably take one question or maybe, I don't know if I went over.
Okay, thank you, Dr. Wang, for your informative talk. Any question from the floor? I have one question.
Sure.
Hold on. Anybody? Yes? Oh, please.
I wonder if there is a possible scenario that is that a successful vaccine producer would authorize a manufacturer in Taiwan to produce a vaccine that works. So in that case, that would speed up the supply, at least for the Taiwanese population. And is that on Taiwanese government's plan?
I don't know what the government's plan is, honestly. They didn't tell me. But if there's a successful vaccine that's proven to be effective and Taiwan could get on it, say we could help you manufacture just like we manufacture masks here. and they'll be like, oh, you could double our production, we'll give you, 4 million, 5 million right off the bat, so you could cover your healthcare workers and the highest risk people. I think that would be really nice. Well, Taiwan continues to develop its own vaccines and build capacity. I mean, basically, you need some sort of vaccine strategy for the next year. And you cannot just do big bets.
Professor Wang, yeah. Okay, yeah, thank you for your wonderful talk. So I just wondering, how about protection efficacy of the mRNA vaccine, and when will approval such kind of vaccine?
Yeah. So I mean, it looks like the first ones that are going to get approval are the mRNA vaccines, and it's probably, relatively safe, and it's it's in phase 3 clinical trials. And so again, if it's an mRNA, you need refrigeration, right? You need the cold chain, and so if you want to bet on that one, Taiwan should prepare for the cold chain. I mean, if you think that's gonna work, right? And so you might want to bet a third on the mRNA vaccine, a third on the protein, and a third on the viral vector, right? And because there are like 11 in clinical trials, is about equal number for each of the technologies.
Okay, because time limit, thanks, Dr. Wang again.
Thank you.
Okay, next speaker is Dr. Stowell. He cannot be here because he will give us the virtual video. And Dr. Stowell, he currently is a associate pathologist in the Brigham and Women's Hospital at the Harvard Medical School. He also the member of the faculty of pathology in the Harvard Medical School. And he will give us the talk is about the diagnosis and the therapeutic utility of IgA for COVID-19. Let's start it.
I want to thank Futan Liu and the other organizers of this meeting for the opportunity to share recent developments in our lab regarding the potential utility of IgA anti-SARS-CoV-2 antibodies in both the diagnosis and therapeutic utility of convalescent plasma in the treatment of COVID-19. Serology has been used for many years to examine infectious disease. This is primarily focused on the diagnosis of chronic infections such as HIV, HCV, and HPV, but it can be used in the acute setting as well. It's also been a powerful tool in establishing the epidemiology of infectious disease, with important caveats regarding the rate at which antibodies form following exposure to an infection, and obviously the half-life of those antibodies once they form, which can be presumably influenced by the severity of the disease and obviously the type of infection. And also, something that's been around a long time, convalescent plasma, is actually a somewhat poorly understood therapeutic intervention that's simply in its design in terms of having antibodies, but exactly what those antibodies need to look like, the titer of the antibodies, whether they need to be neutralizing, and the isotypes that should be present or may convey therapeutic potential for this particular product really have remained largely unknown. And so we decided to develop a particular serological assay, actually in collaboration with the CDC. And when we're doing that, obviously you want to identify an appropriate target. And the reason this is important is that this can dictate in some ways the sensitivity and specificity of the assay, depending on how similar the target may be for one infectious organism to other infectious organisms, and also whether the target's a common target of the immune system with respect to forming antibodies in this particular assay. It may be the assay is designed in such a way that it can provide information beyond simply whether a patient has antibodies against a particular infectious organism, with respect to whether that antibody profile may have some correlates to actual immunity. In other words, do the antibodies we're detecting correlate with neutralizing antibodies, for example, or antibodies that function in such a way that could be sterilizing in terms of immunity, or at least protect someone from reinfection if they have established naturally immunity from an infection. The detection system is obviously important because this conveys analytical sensitivity and specificity, whether it's a point-of-care test or an ELISA platform or somewhere in between. These types of systems have inherent limitations and advantages to them that really are important to consider when trying to develop serological tests as they are with any type of laboratory test. And then also the practicality of developing the test. So whether the test can be done very quickly, how many full-time employees are required to do the test, what the turnaround time of the test, all that influenced the cost and just the feasibility overall of actually executing the test in a clinical lab environment, and whether it has sufficient turnaround time to actually be useful and guiding therapy. So everyone's, I think, familiar now with SARS-CoV-2, the virus, and there's various antigens on this virus that potentially serve as targets when trying to develop a serological test. Many tests use a nucleoprotein found inside the virus. Some use the matrix protein, but most have used a spike protein to look, once again, at a target that could be used to determine whether someone's made antibodies. We decided to focus on the receptor binding domain specifically within the spike protein, in part because several studies early on, these studies were really being developed in early April as the pandemic was starting to really ramp up. It had suggested, at least with SARS-CoV-1 and MERS and other coronaviruses, that this domain may be responsible for interaction with host cells. In particular, this virus, it was suggested this interaction may be responsible for no interactions with ACE2 that, once again, may be important for getting into the cell. And so we thought maybe examining antibodies against this particular domain may be important in establishing potential function in terms of it, once again, being a correlate with neutralizing antibodies. And we also wondered whether these particular domains may be helpful in establishing specificity for actual infection to SARS-CoV-2 versus other coronaviruses. And of course, we needed to know about sensitivity, and we needed to explore, of course, when patients were PCR positive for SARS-CoV-2, did they go on to develop antibodies that could be detected against this domain? None of that was known when we started these studies. So developing serological tests usually takes many years and many samples. This is something that's not normally undertaken in several months, especially in the middle of a pandemic. But once again, this was something that the Center for Disease Control reached out to Emory University, asking us to develop a test, in large part early on, to actually screen their employees before a lot of the PCR-based tests and antigen-based tests we use very routinely now were widely available. So this was just one of the early efforts, along with many efforts to try to survey individuals in the population to reduce the risk of spreading the disease. And so we elected to use an ELISA platform simply because we had the clinical infrastructure to be able to do this assay. I think everyone's familiar with an ELISA. It's a very simple assay in many ways. Basically, there's some type of antigen coded on a plate. In this situation, it was a receptor binding domain, a SARS-CoV-2 spike protein, and then, obviously IgG or IgA or IgM or other isotypes presumably could bind to the domain present coded on the well, and then secondary antibodies specific for each of these isotypes are then used, conjugated with an enzyme, horseradish peroxidase in the situation, and then there's a substrate that's converted to a color that can be read out as some binding event occurring. And so to do this, we initially took a cohort of PCR positive patients confirmed in the hospital and basically did a serial dilution assay using historical negatives that were collected for other purposes pre-COVID as a control. And basically the goal here was to simply determine whether we could find a one dilution assay that would be a pragmatic approach to doing this clinically. The goal here was not to do endpoint titers because we felt like many labs felt the same way. It just wasn't going to be practical to try to develop that in the clinical setting. So these are results just showing different titer dilutions with different secondary antibody concentration we use to optimize the actual concentration of antibodies used in this particular assay. Once again, to reorient everybody to the infection, and presumably the virus binds to epithelial cells that are decorated in ACE2, and that antibodies that form against these particular domains on the surface of the spike protein are thought to be, at least early on, were thought to be in part responsible for the ability of these antibodies to neutralize the virus or prevent it from infecting host cells. And so we wanted to determine whether antibodies against the receptor binding domain actually correlated with neutralizing antibodies. And therefore, I want to say this very carefully, that the goal here wasn't to develop a complete surrogate for functional antibodies. And frankly, we don't know if neutralizing antibodies actually convey an immunity, because a lot of the antibodies are collected in the serum. And it's really not known what's at the mucosal surface, which is where the virus is most likely to infect people. But we felt like this would be potentially useful to at least produce data, or at least determine following doing experimentation whether there's some kind of correlation here. And so to do that, we simply, this is work done by Mehul Safar and Jens Rammert, did a neutralization titer analysis, compared it to titers using RBD-specific antibody analysis for IgG, and found there's a really good correlation, suggesting that these antibodies may be functional. I mean, it doesn't demonstrate this, of course, but may provide a very quick surrogate for functional antibody analysis. The next question, of course, we want to determine is whether these antibodies were specific to SARS-CoV-2. Once again, this is important because someone had a cold virus exposure or had longer lived memory from a previous cold virus exposure, that could complicate our ability to use this in a diagnostic fashion or even in epidemiological survey fashion to determine whether people had really been exposed to this particular virus, SARS-CoV-2. And it's important to note here that the percent identity between SARS-CoV-2 and common circulating virus, in particular HKU1 and OC43, was quite low, less than 9%, 10% identity. So these results suggested probably directly because antibodies are targeting these domains, and therefore there's selection questions that probably drive some of this diversity. That the cross-reactivity may also be low, but we obviously wanted to test that, and so we looked at specimens from patients who had been diagnosed with COVID via PCR test and asked whether antibodies that were reacting with COVID also reacted with HKU1 or OC43. And we saw for the large part, these antibodies did not cross-react. There's very little cross-reactivity, suggesting that this assay on the RBD as a potential target may be useful and provide some specificity to the diagnosis of SARS-CoV-2. So next, we want to determine the sensitivity in patients who are hospitalized for COVID that were PCR confirmed with respect to how many of these patients developed antibodies against the virus. And once again, this was not known early on, and to what extent that occurred over time. So what are the kinetics of antibody formation? When we typically think of antibodies forming against any antigen, we typically think of IgM coming up first, and IgG, especially if it's a naive immune response. Because we didn't see any antibodies forming or present in controls, we presume that we would see a naive response, although there obviously could be memory and evidence of antibodies, so they were below the level of detection. But be that as may, this is kind of what the hypothesis was at the time. I don't think we were alone in thinking this. And to be fair, there's always going to be a window period with any infectious disease testing. It's quite large with serology, but it's a little smaller with NAT testing. And so we want to determine kind of what this window period may be when patients present with infection. And so what we did is look at IgM first, and what we found was that within a few days, several of the patients, quite a few actually, especially if they're PCR tested, a few days of hospitalization, and with a few more days, 5 to 9 days of symptom onset, IgM against the RBD of SARS-CoV-2 could be detected. And this is also true of IgG, where very similar kinetics were observed. So these are exact same samples tested for IgM or IgG. And we're surprised to find a very similar kinetic picture with IgM-Ig forming against this particular domain of SARS-CoV-2. Once again, suggesting there wasn't this kind of classic IgM to IgG class switching. This has been shown by many other labs now. So we did a receiver operating curve analysis to determine the specificity and sensitivity of these tests, and found that IgM behaved really well, actually, looking at every sample that came through the door of PCR positive patients irrespective of time. And it performed a little better than IgG, which is not entirely surprising given the fact that IgM's thought to come up a little earlier. You can kind of see that here. But overall, they seem to perform fairly well, and as I'll show later, perform with greater sensitivity and specificity over time, which is not unexpected. Most serological tests look at IgM or IgG or both. Very few look at other isotypes, but given the fact that this is a mucosal infection and there are a variety of different antibodies, including IgA, which is specialized in its ability to be bound by the poly-IgG receptor, which transcytoses the antibody across the respiratory epithelium into the lumen of the lung and other mucosal surfaces. We want to know what serum IgA levels would look like in these patients, and whether there may be differences in IgA level, in part because we want to know if there are serological correlates with disease improvement that we could use in convalescent plasma to try to characterize early on what factors in convalescent plasma serologically you should be looking for to pick units that may be the most effective. Once again, not really knowing what would be the most effective, but hypothesizing at least the IgA may be important in this regard. So we next looked at IgA, and we were surprised to find that IgA levels formed early on in infection looked to be quite specific. In fact, when we did receiver operating curve analysis, the AUC area under curve for IgA against SARS-CoV-2 was really favorable, much more favorable actually than IgG and IgM, which was not at all what we expected. And so these results suggest actually as a diagnostic maneuver, examination of IgA antibodies against SARS-CoV-2 could be really powerful. And this is examining the data stratified by time post-symptom onset. So of course, as patients have an infection for a longer period of time, the presumption is that antibodies are going to be more likely to form, and that's exactly what you see. But even when you stratify it by days weeks post-symptom onset, you can see that IgA performs better than IgM and IgG, and it's really apparent both at week 1 and week 2. So the likelihood of picking up infection based on serological test alone is much more powerful for IgA than either IgM or IgG. This is not only true of post-symptom onset data, but also post-PCR data. So when patients walk in the door or maybe seen at an outside clinic and we're given a PCR test via nasopharyngeal swab, and then we obviously examined over time, looking at the serology, IgA outperformed IgM and IgG. So once again, not a common isotype used in clinical assays for any infectious disease, but for whatever reason, this infection, it seemed to perform very well. We next want to determine whether there was any correlation between disease severity and the development of antibodies. At the time, we weren't quite sure whether patients that did more poorly were more likely to not make a productive immune response. And therefore, we decided to look at the data based on stratification with respect to whether patients had been intubated or not. And what we found was that patients who were intubated actually were more likely to make a robust immune response than those who were not. So these results suggested that at least at these later time points, and these data were censored based on 2-week data so we could compare them equally because patients who were intubated were in the hospital longer. This is looking at similar time ranges post-hospitalization. So those patients who were doing really poorly were making a robust humoral immune response. Obviously, you know what the CD8 T cell response looks like in these patients. And once again, when we did ROC curves on these patients, those patients who were intubated had more favorably performing ROC curves with respect to sensitivity and specificity. Once again, suggesting that serological examination of the patients in this ICU setting may be a useful strategy, especially if you have a patient who looks like they have COVID, but for whatever reason tested PCR negative initially upon presentation. And these data just show the AUC curves, showing that once again, if a patient's intubated, they have more favorable behavior than those who did not. Once again, we don't know the kinetics or where these antibodies come up after, and one of the reasons the patients get sick is because they don't make an early immune response or it's delayed in some way. And so we don't know any of those types of data right now. These are just the cross-sectional analysis of these data. The next one to determine whether patients who were so-called patients under investigation in the hospital were more likely or capable of being diagnosed with COVID using serology that were initially NAT or PCR negative. And the reason this was important is we saw a lot of patients as we started doing chart review that looked like they had COVID. They had kidney injury, they had a troponin leak, they had liver function test elevation. They had a lot of the common low-grade end organ injury these patients typically present with, obviously respiratory compromise, but did not test PCR positive. And of course, NP swabs, it's a heterogeneous mixture of material that's obtained when an NP swab is done and not as homogeneous as blood. And so you want to look at this, and what we found was that there were a fair number of patients who did test positive serologically. And when we examined these patients over the course, they clearly had seroconverted. So these results suggest that serology may be useful, especially in the hospital setting, when patients present and for whatever reason have a negative PCR test, that within a week or two, if they still look like they have COVID, they really should probably get a serological test. And the reason this is important, of course, early on, there wasn't a specific treatment modality. The remdesivir trial was going. No one really knew if it was going to work or not. And so it was mostly supportive care. That's certainly changed. So remdesivir obviously is a common approach given decadron or dexamethasone is very common in the ICU. And so there's and obviously common with plasma, which I'm going to talk about in a second, are specific treatment modalities where the diagnosis is really important to make accurately. Furthermore, a lot of these patients were on respiratory isolation and therefore put healthcare workers at risk. In fact, we found that early on, 9% of the healthcare workers had seropositivity, suggested there was some exposure occurring that they weren't aware of. Most of those healthcare workers were not symptomatic. So I'm going to shift gears to convalescent plasma and talk a little about some of the studies we've done in this area that are similar to what I've just talked about with respect to diagnosing COVID-19 serologically. So this was a paper published in JCI by Mike Joyner's group looking at the potential safety. So this was just a non-randomized prospective observational study done in many sites within the US. And basically what they found was there was really no evidence that they could detect of severe transfusion reactions, essentially following use of convalescent plasma. And to be fair, that wasn't really known or hadn't been studied at this level prior to this. And so I think these data were important, and these data have been followed up with serological evaluation of convalescent plasma. So these units were never characterized before transfusion. Segments of these units or material from the units were saved and sent off to the Mayo Clinic to be tested. And what they found is when they looked at antibody levels, the higher dose and the higher titer antibody seemed to correlate with efficacy of convalescent plasma, especially if it was given early on. And this is, I'm just going to share a case of a patient we had early on that had, well, I'll just share the case. So it's a 3-week-old infant with trisomy 21 and congenital heart disease. The patient presented with decompensated heart failure due to this congenital heart disease. And because of that, was noted to have respiratory compromise that required intubation. So the patient was awaiting surgical correction of an AV canal, atrioventricular canal, but was really having a difficult time overcoming this respiratory compromise. And it was noted on hospital day 5 via MP swab that the patient was positive for SARS-CoV-2. And the patient remained positive for a while. It wasn't clear what the role of remdesivir should be in infants and pediatric patients in general, but it was believed that it may be helpful. So it was given for a 14-day period, and the patient didn't seem to respond. And so kind of as a last-ditch effort, this patient received convalescent plasma from 2 units from one donor, or 2 doses, I should say, from one donor, and 2 doses from another donor. And what we found was that shortly after exposure to the first donor, there was clearance of the virus, suggesting that something may be going on with respect to just giving a timing with convalescent plasma. And so given the fact that we had the test, we decided to look at the donors. What we found was that the first donor had higher titer IgM, IgG here in red, and IgA. The second donor had very low antibodies here against IgM, IgG, and IgA against the virus. What we found very interestingly is that when we looked at IgM antibodies or IgG antibodies, pre and post here in blue, nothing really changed for IgM and IgG. But IgA anti-RBD antibodies changed significantly following transfusion, and these levels were sustained over time, and obviously correlate with the clearance of the virus. It really made us start thinking about the potential role of IgA, not only in diagnosis, but the utility of convalescent plasma. If there's a lot of inflammation going on, presumably there's enough leakage into the lumen of the respiratory tract so that IgG and IgM could get across, but obviously IgA, once again, has this unique ability to get across regardless. And so we decided to look at this in convalescent donors in general, based on IgG, IgA, and IgM levels. We found that while there's some correlation between IgG levels, which are the common isotype detected in blood centers in the country, in the US at least, the correlation wasn't perfect, of course, and we looked at donors that were completely IgG negative. A lot of these had IgG levels, shown here in red. And so these results suggested that the way we were characterizing units may miss on the potential influence of IgA. And this is just showing general trends, but a fair degree of lack of correlation between IgA, IgG, and IgM, which is not too surprising actually. But once again, started begging the question whether these particular features of convalescent plasma units should be considered. We looked titering now over different hospital days and found that IgG and IgM levels typically were very detectable within 14 days, especially after 10 days of hospitalization. But the IgA levels were variable. So these levels are low, but it could be detected based on our single dilution assay. So this would be helpful in a diagnosis. But the titers are very low. So the single dilution assay is not telling us titers. It's just an alphanumeric number actually based on an OD cutoff. These patients had very low levels, suggesting that some patients may particular benefit even in later stages of hospitalization if they had corresponding low levels of IgG antibodies against SARS-CoV-2. We next wanted to know how stable these antibody levels were in convalescent plasma donors. So to do this, we looked at donors who had donated more than once as a repeat donor. We found that IgG levels were generally quite stable in repeat donors, but IgA levels and IgM levels decline on average. And these are the data here. You can see the IgM and IgA levels decline over time. And once again, because this is the orthovitreous instrument, this is the instrument used in most of the blood donor centers here in the US, there really wasn't a decline observed with the orthovitreous, which once again correlates usually quite nicely with IgG, but would not see a potential decrease in IgG and IgM. And I'm not suggesting that these antibodies are the only thing that can be beneficial in convalescent plasma. They may not actually influence at all. We don't know. We only had the one case that was correlative for sure in nature. But these results just suggest that these antibodies may be changing differentially over time, and that they are important. Considering these potential differences may be important. And finally, we actually looked at potential correlations between IgG, IgA, and IgM levels against the RBD with the orthovitreous instrument that's once again commonly used, and found that IgU levels correlate pretty good. It wasn't quite as tight as we expected. The IgA levels and IgM levels really failed to correlate well at all. So these results suggest that once again, the way we're characterizing units may be useful. I think it's better than nothing with respect to determining whether we're shooting a blank. In other words, convalescent plasma may not have really any antibodies in it versus having something in it, but that maybe a more detailed analysis of the antibody composition may be useful when trying to determine whether a particular unit may fill the humoral immune response deficit in a particular patient. So looking at the patient and the unit to determine whether we can create a combination, as we do for other blood products, that actually fills the gap in this particular patient's humoral response, whether it's G or A or M against SARS-CoV-2. So I hope the results I share with you today suggest that IgA may be a useful serological tool when seeking to diagnose patients with COVID-19, particularly in the hospitalized setting, which is, once again, not a common approach we use clinically, but given the severity of the disease, the degree to which it can be spread, and the recently highlighted therapeutic interventions that can be used to treat this disease, we feel like this is at least something to consider. Obviously, it needs to be studied in more detail. But we were surprised by these findings, and that's why we're sharing them so that they can, once again, consider future studies. And also this idea that tailored CP therapy may be useful. Early on, people were understandably using, criteria based on whether people have been infected or not, because the serology tests were not widely available. And then they've become more widely available, which is fantastic. And the idea we're proposing is that an additional layer of understanding regarding both the convalescent plasma unit and the patient, and whether the combination of the two really matches well to fully fill the gaps that may be present in that patient with respect to antibodies they're forming may be useful. With that, I'd like to thank my lab and all the people in it. Connie Arthur was unbelievably involved in these studies in terms of collecting specimen. Hans Berkey, which is a student in my lab who's been absolutely wonderful. It's not in this picture. He's a new addition to our lab. He did the lion's share of the test development and analysis of all the data I showed today. Just an unbelievably great person to have around, and just been a wonderful colleague in all these studies. And then a whole host of people that were literally involved in every aspect of these studies early on in the pandemic, from medical technologists in the lab who acquired specimens for us, to other people in the immunology lab to help develop the test in the CLIA certified environment, to Hans, who I mentioned earlier, who's been super amazing, and Connie Arthur, who led a lot of the aliquoting and biopository storage. And with that, I would normally take questions, but I think this is a recorded video, so I'm grateful once again to be here, and you're more than welcome to reach out to me via email if you have any additional questions. And with that, I appreciate the opportunity once again, and thank you very much.
Because doctors are not online to answer the question, so let's move on to the next speaker. Our next speaker is Dr. Sytwu Huey-Kang. He's the Vice President at the National Health Research Institute. He's also a distinguished investigator at the National Institute for Infectious Disease and Vaccinery at NIH, and he received his MD from the National Defense Medical Center and PhD from Stanford. His current research focuses on the autoimmune disease mechanism of insulin-dependent diabetes. Let's welcome Dr. Sytwu.
So thanks for the invitation and such kind introduction. Today I'm going to present some of our recent progress about the development of vaccine and drugs against the SARS-CoV-2 at the NHRI, the National Health Research Institute, which is a mission-oriented research institute and supported by Taiwanese government. At the beginning of this presentation, I would like to give a very, very brief overview about the global SARS-CoV vaccine landscape. So according to the data from the current issue of Nature Review Immunology. There are more than 160 vaccines around the world. So most of them are still in the preclinical development, but among them, there are 27 vaccine programs are currently under clinical trial from phase 1 to phase 3. So basically, there are 6 major platforms and a strategy for those vaccine programs. More than 1/3 are based on the protein subunit-based vaccine development. So this kind of strategy require the adjuvant, and the biosafety issue is relatively okay, but the major concern is the immunogenicity may not be strong enough to make good protection against SARS-CoV-2. So around 30% vaccine programs are based on the viral vector approach. So mainly the adenovirus vector and some other like measles and the influenza vector. So around 25% approach are based on the nucleic acid vaccine development, including the messenger RNA vaccine and the DNA vaccine. And some of them are whole virus inactivated approach, and a few of them are live attenuated virus. So each strategy or each platform has its own strengths and advantage, but also has its limitation and shortfall. So compared to protein subunit-based vaccine approach, those viral vector-based strategy can relatively induce strong immune response, including the antibody response. Oh, I guess I can use the laser point instead of the mouse. So basically, there are different kind of adenoviral vector. All of them are non-replicating viral vector, including the adenovirus human serotype 5 or human serotype 26, and also the chimpanzee-based adenoviral vector. And some other vectors are based on the vascular stomatitis viral vector, and the other are measles or influenza virus. So basically, those viral vectors can induce pretty strong immune response, including the neutralizing antibody, the Th1 immune response, and the cytotoxic T lymphocyte, or some of them, if through the respiratory mucosa vaccination route, can actually induce strong residential memory T lymphocyte in lung. However, there's a major concern, especially for human adenotype 5 or 26, is the pre-existing antibody, so that will negatively affect the antibody production and also the cytotoxic T cell immune response. So the chimpanzee-based adenoviral vector designed by Oxford University and AstraZeneca can certainly avoid this kind of shortfall. However, those immune response based on the parental design, they are intramuscular injection. So lots of experimental approach based on the respiratory mucosa vaccination route are still needed. And the other thing about the measles viral vector base or the influenza viral vector base, there are still extensive biosafety concern, especially the potential recombination of live attenuated influenza viral vector in lung. So they are DNA-based or the RNA-based vaccination strategy, like Moderna. So Moderna, the vaccination can actually induce both Th1 and Th2 immune response. Depends what kind of adjuvant they use. For example, the aluminum can actually induce the quite strong Th2 immune response. That would be a major concern to induce the antibody-dependent enhancement to its adverse effect for this aluminum-based adjuvant in RNA-based vaccination strategy. For Inovia, they developed the DNA-based vaccination. Again, both DNA or RNA-based vaccination strategy, they require the adjuvant. And the live attenuated virus, that's still extensive biosafety concern for the potential recombination. So this is the inactivated viral vaccination strategy, can induce both Th1 and Th2 immune response. Again, depends on what kind of adjuvant they choose. However, this kind of approach cannot induce strong cytotoxic T lymphocyte. So in early of this year at NHRI, based on some facts and previous experiences, we quickly mobilized our research team in National Institute of Infectious Disease and Vaccinology. So led by our director, Dr. Liao Jinlun, and his research team. So in NHRI, we actually have a bioproduction plant. It's small scale, but under the standard of PIT GMP standard. And also in past 15 years, we were trying to help some other domestic vaccine company to build up the clinical trial for flu and avian flu and the EV71 vaccination program. So based on those facts, so our director, Dr. Liao, and our research team quickly developed 4 different platforms in NHRI, including the synthetic peptide-based or DNA-based, adenoviral vector-based, and the protein subunit-based strategy. So as I mentioned previously, each platform or strategy has its strengths, advantage, but also has its shortfall. But sometime around May, I guess 3 domestic vaccine companies, they all focus on the protein subunit vaccine. So I think NHI under the mission-oriented strategy. So we don't want to compete with some other company in Taiwan. So we stopped our protein subunit project, and we really focus on DNA vaccination strategy. So each platform we actually selected from different candidates. For example, for the synthetic peptide-based vaccination strategy, we selected from 20 candidates and pick up the best one. And for DNA vaccination, we selected 1 candidate from 6 potential targets. And also the adenoviral vector, we select 4 candidates, one target from 4 candidates. For protein subunit, we select 1 target from 8 candidates. All this selection was screened by antibody titer, neutralization capability, and animal-based protection. So for past 3 months, we really focused on the DNA vaccination development. So we pick up the potential candidate vaccine, encode the SARS-CoV-2 spike protein, and compare the other DNA vaccine actually encode the SARS spike protein. we immunize mice with these two different vaccine candidates, and 4 weeks or 6 weeks after vaccination, we pick up the serum from those vaccinated mice, then check their antigen-specific antibody titer. So from those ELISA assay, we actually coating different antigen, including the full-length SARS-CoV-2 spike protein and the receptor binding domain only antigen, or the S2 domain antigen. So both DNA candidate, for example, encoding the COVID-2 or the SARS spike protein, can induce pretty strong antibody response against the full-length COVID-2 spike protein. But for antigen coating the receptor binding domain, only the SARS-CoV-2 spike DNA can induce specific antibody recognize the SARS-CoV-2 receptor binding domain. However, for the S2 domain, both DNA vaccination can induce equal strong antibody titer recognize the S2 domain. So from this result, we actually support our DNA vaccination can induce strong specific antibody recognize the SARS-CoV-2 spike protein, and the antigenic differences is among the receptor binding domain. So the S2 domain actually quite conserved between the SARS virus and the COVID-2. So to further address those spike-specific antibody can really neutralize the virus infection. So we culture the viral E6 cell and infect those cells with SARS-CoV-2. Meanwhile, we add the serum collected from either immunized from SARS or SARS-CoV-2 DNA vaccine. So apparently, the serum collected from COVID-2 vaccinated mice can present very strong neutralized capability against the viral infection, and the titer is even higher 6 weeks after the vaccination. So we are also trying to establish an appropriate and available animal model for testing our vaccine. So according to Dr. Yan in previous section, she already mentioned the mouse ACE2 apparently is not a good receptor to interact with the COVID-2 virus. Instead of the human or bat or the swine or the cat, they all express the ACE2, which can interact with COVID-2 and enhance their infectivity. So based on the schematic strategy set up by the US FDA animal efficacy rule, so there are several requirements for the animal model to meet the FDA efficacy rule. The first requirement is the viral replication. The second requirement is clinical disease, and the third requirement is the pathogenesis. So apparently, mouse is not a good model to fit this rule. So we go back to look at the amino acid sequence between different species, including human, African green monkey, and the macaque, and the ferret, and the mouse, and the hamster. We look at the amino acid sequence between the critical domain to interact with the spike protein. The mouse amino acid apparently are quite different from human. And the hamster only 2 amino acids different from the human sequence. So based on this result, we test the hamster as an appropriate model to test our vaccine strategy. So the hamster were inoculated with different virus titer, and 3 or 4 days after inoculation, their body weight decreased dramatically, very significant. So based on this, the virus apparently can affect the hamster at least in the change. And the other analysis, we pick up the lung after the virus inoculation, then look at the virus load. That's lung pick up day 3, so after inoculation. So there are different input from the viral load, and we look at 3 days after inoculation, there are at least 1,000 to 10,000 amplification of virus replication. So we use the hamster to test our DNA vaccine. So the hamster received 2 vaccination of our DNA vaccine candidate, and 4 weeks later, we challenge it with the virus. 3 days later, we sacrifice the mice and look at the body weight change. So basically, the mice vaccinated with the DNA, they are protective from the body weight loss. Their body weight is gaining at a regular way. And also, we sacrifice the mice and look at the viral titer. We assay by the TCID50 analysis and found the hamster vaccinated with the SARS-CoV-2 DNA vaccine, the viral titer is significantly decreased. So I apologize some Mandarin on this PowerPoint. So in past 3 months, we built up our research cell bank and complete our master seed bank. Now we are working on the working seed bank and also doing our characterization of this cell bank. We also start the pilot production in the GMP condition to produce some DNA vaccine for the preclinical toxicology test and the GMC test. So if everything is going well, we will try to file the IND end of the November and trying to start our phase 1 clinical trial end of this year. So now we are moving to another story about the molecular antibody generated against the SARS-CoV-2. So the story should go back to 17 years ago during the SARS outbreak. So the NHI collaborated with the Institute of Preventive Medicine, National Defense Medical Center. We produced a whole panel of molecular antibody. Those antibody can specifically recognize the SARS spike protein, and also they can neutralize the pseudovirus-based infection for the viral cells. So end of the January, the first Nature publication present the whole viral genome. We actually look at the original antibody we produce. They recognize the SARS antigenic epitope is located from amino acid 1146 to 156. That's antigenic epitope recognized by those antibodies. So we are very fortunate. The amino acid sequence are completely identical between the SARS-CoV-2 and the SARS. So we assume those antibodies can specifically recognize the epitope of the spike protein of SARS-CoV-2. So the first strategy is trying to establish the rapid diagnostic kit. So we generate the prototype detection kit by coating the antibody originally recognize the SARS. So this prototype detection kit can readily detect the viral lyset from SARS, can also detect some different strain of COVID-2 cell lysate. And for other human coronavirus, including the 229E or OG43, they don't react. And this antibody also do not react to other influenza viruses such as H1N1 or H3N2 or influenza B. And the specificity of those antibodies is quite good, but however, the sensitivity is okay because we detected the lower limit is around 50 to 62 nanogram per ml. It's okay, but not good enough. So we are trying to enhance the sensitivity for the rapid diagnostic kits. We were collaborating with some international semiconductor manufacturing. They designed some so-called BioFET biosensor system, either to detect the IO-sensing system or genotyping system or immunoassay system. So we coated our molecular antibody on this biochip and then do another test. The sensitivity can increase at least 1,000 to 10,000. So we also test the therapeutic potential of this molecular antibody. So the hamster will inoculate the virus first, and we inject those hamsters 3 or 24 hours after the virus inoculation. So 2 different kinds of molecular antibody we use can really protect the hamster, at least the body weight did not change compared to the control group. And also we sacrifice those mice, detect the viral titer in their lung. The hamster received the 1 shot of therapeutic antibody, their viral titer decreased significantly. We also trying to establish another mouse-based animal model for the SARS-CoV-2 research. So it's based on the amino acid differences between mouse and the human. So we use the CRISPR-Cas9 genome editing strategy to change the 5 amino acid to make the beta interaction of this mouse-based, the ACE2 receptor to get the beta interaction with the spike protein of COVID-2. So this mouse model is currently under test right now. So now the final part of my presentation is the drug development in NHRI. So the first story is about the synthesis of remdesivir. So the story will go back to the 2005. During that time, the avian flu outbreak. So Taiwanese government cannot really order the Tamiflu from Roche. So at that time, 15 years ago, so Taiwan government initiated an emergency action, and NHI was assigned to synthesize the Tamiflu. So NHI spent 18 days to synthesize the Tamiflu, and based on this result, we get better negotiation with the Roche to get a safe amount of the Tamiflu. So in this COVID-2 pandemic, the NHI will also trying to synthesize the remdesivir. So we start to do the synthesis activity on February 5th and spend 2 weeks. We complete the remdesivir synthesis in milligram scale. And 5 days later, we upgrade the synthesis to the gram scale. And also we recrystallize the 1 critical step for the kilogram scale of synthesis step. However, the Taiwan government collaborated with the Gilead company, and Taiwan joined the multi-country clinical trial with Hong Kong, Korea, Singapore. So basically, we stopped this the following process for the remdesivir synthesis. The second part, this is the schematic presenting the synthesis process, and we also finished the technical report on the whole process for this remdesivir synthesis. The second part of our drug development is based on the artificial intelligence approach. So we collected two different learning database. One is consists of the compound reported or proven active against the SARS or the SARS-CoV-2 or HIV. And then the other database, actually, we collect all the known or published 3G-like protease inhibitor, and we test in our screening model, we call the FIP virus assay, the feline infectious peritonitis virus assay. So this assay, we can easily do the large scale screening. Then all of the results will feed back to the AI learning system, and we collect all of the information based on our compound library and also some old drugs. So this is our screening strategy. So we pick up 254 old drugs or the pharmacologically active compound. Then first screening by the feline infectious peritonitis virus and pick up 23 hits. Then we further assay by the human coronavirus OC43 screening system and pick up another 15 hits. And now we are doing the P3 label, the SARS-CoV-2, the assay system, including the TCID and the plaque reduction assay system. So this is one example. We pick up some drug from the repurposing old drugs, including some anti-parasite drugs, and also some hydroxychloroquine and its analog. And also we pick up some remdesivir active metabolites from our screening system. And this is some other compounds we pick up through our screening system. So this is one of the examples. We look at the new drug development. It's a new compound synthesized in NHI. We call the BPR3C0001. So this compound can show very good inhibition curve, and the selectivity index is more than 120. And this is another story. We actually developed some drugs against the COVID-2 that's based on the 17 years experiences. During this SARS outbreak, some of our colleague, Dr. Lee, and her research team actually developed some synthetic compound derived from tyrofluorine. And some of the analog can actually show very strong inhibition against the COVID-2 if compared to the remdesivir here. So one of the analog derived from the tyrofluorine can actually show much potent if compared to the remdesivir. And also this tested by the other human coronavirus 229E, again, one of the analogs show very strong inhibition efficacy. So this is also done in the P3 level lab. We actually tested the viral E6 cell infected by SARS-CoV-2. Again, some of the analog, especially this one, DPQ33B, can show very, very strong efficacy for the inhibition of viral infection. So this is the table summarizes some of our drug screening. Okay, I think I will stop here and take the question. Thank you.
Okay, thank you, Dr. Sytwu. Because time limit, we allow one question, if any question for Dr. Sytwu? Yes, Dr. Wang.
Thank you very much, Dr. Sytwu. I have one question about the use of rapid antigen test. It looks like the United States is going to try to reopen schools and businesses early next year with these rapid tests. And this morning we heard a lot about the different strains of the SARS-CoV-2 virus. And has anybody done a systematic study on these rapid tests and whether, how we should look at the data? Because, if it's positive versus negative, and it shows up with the slightly different, as your data showed that some is darker than the other in these rapid tests. And how do we use these tests across the different strains?
Okay, so based on our own results, I guess the specificity is quite okay because our antibody can recognize only SARS and the SARS-CoV-2, not for other human coronavirus such as OC43 or some other 229E, and not others respiratory syncytial virus or some other influenza virus. But the sensitivity actually is not good enough. So we are trying to do the biochip assay system, trying to increase their sensitivity. However, those are preclinical assay. For example, in Taiwan, based on the regulation set up by the FDA and the CD in Taiwan government, if the rapid diagnostic kits can get the license, that's based on you have to done certain cases of the clinical sample. But in past a few months, I guess the case number in Taiwan is quite low, so it's somehow difficult to validate those detection kits by the real clinical sample. I guess that's one hurdle for the overall development for those rapid diagnostic kit in Taiwan.
Okay, let's thank Dr. Sytwu again. I'm going to move on to the next speaker. Okay, our last speaker is in this section. It's Dr. Hsueh-Fen. He right now is a distinguished professor in the Department of Life Science and the Graduate Institute of Biomedical and Bioinformatics at the National Taiwan University. And he has used the bioinformatics and big data analysis to study the repurposed drug against the SARS in 2003 and now in 2020. And let's welcome Dr. Wang.
Okay, thank Professor Lee's introduction. And I also want to thank Vice President Dr. Liu and the organizer committee for inviting me here to present our recent work on drug repositioning for COVID-19. So as you know, the confirmed cases of COVID-19 disease are more than 32 million. So it's important to develop the vaccine or the drugs against the COVID-19. So from the literature search, from the PubMed literature search, we use COVID-19 as the keyword. Then you can find more than 58,000 literatures have been reported. And from the clinicaltrials.gov, you can find that there are more than 3,000 clinical trials ongoing. So traditionally, we found drug discovery is through serendipity. It means discovery by accident. So for example, the cow ate the sweet clover and got the sweet clover disease, which is bleeding to death. So the Dr. Link isolated compounds called warfarin, which has the anticoagulant effect. So the other famous findings including penicillin. So to shorten the drug development process, so drug repositioning is the good strategy. So it is a rapid and cost-effective way to discover new therapy options for emerging disease such as COVID-19. So there are two approaches. One is from known compound to new target. So the other is from known target to discover new indication. So in 2003, I joined the national team led by Professor Wang. We used drug structure similarity for drug development. So at that time, some drugs have been no effect against SARS. So we used data mining to search the similar structural compounds. Then finally, we found more potent drugs as shown in the right side. So in the beginning of this year, Moss invited me to present our strategy. So at that time, I showed that we use two strategies for drug discovery against COVID-19. So one is virtual screening, and the other one is using big data analysis. So we can validate the results using enzyme assay, cell-based assay, and also the animal model. So if you want to redesign the novel drugs, you can also use the drug-drug similarity to find out the core structure of the effective drugs. So how to select the molecular targets of SARS-CoV-2? You know, spike protein bind to the ACE2 on the host cell membrane. Then the TMPRSS2 cleavage this interaction, then the virus enter the host cells. So after the virus enter the host cell, there are many proteins involved for viral reproduction. So such as PL protease, M protease, RDRP, and also the M protein. So our first strategy is using a virtual screening. So if you know the protein structure, you can directly perform docking simulation with the drugs in the compound database. If you don't know the protein structure, you need to first perform homology modeling to predict the protein structure, then further perform docking simulation to find the drugs. So at first, we construct a drug database against SARS-CoV-2. We call the database SARS-CoV-2. So you can access to the URL shown here. So we use 7 drug targets, such as 5 from viral proteins, such as spike protein, RDRP, 3CL protease, PL protease, and N protein, and 2 from human protein, such as ACE2 and TMPRSS2. So our database provides not only the docking score, but also provide the docking structure, ligand information, experimental data, which are retrieval from the literatures. So first, we use the Opal barbell to convert the 3D structure of the drugs, then perform the docking simulation using AutoDock VINA. In the database, we also provide a heat map visualization and the candidate drug list. So we compare the determined 3D drug protein structure. So as shown in the figure, our prediction is good because these two compounds, these are the same compounds bind to the similar residues on the proteins. So users can download our data. So the data contains proteins, ligands, and also the ligands with all poses. We also provide open scripts and the source code on GitHub. So if you are interested in some drugs or proteins, you can perform docking simulation using our source code. So the second part, I want to introduce you how to use big data analysis to find the antiviral drugs. So in the early infection phase, a potent host response suppress the viral replication and leads to the low symptoms, low inflammation, and have a good prognosis. If the host defense mechanisms are defective, will lead to a high viral reproduction and hyperinflammation, high severity, and finally death. So coronavirus stimulate the production of pro-inflammatory cytokines and also the type 1,3 interferons. So the secret interferon 1 and type 3 induce hundreds of antiviral effectors over the interferon stimulate genes to achieve a cell intrinsic state of viral resistance. So this figure shows low viral load induce early interferon response and rapidly viral clearance. If high viral load, the interferon response will be delayed. So the delayed interferon response causes viral persistence and the inflammation, finally become severe disease. So from the literature's reports, we propose a hypothesis. The compounds enhance the host response to type 1 interferons may be effective against SARS-CoV-2 infection. So we use a gene expression-based approach to make such predictions. So here show our schema. First, we need to identify the interferon stimulate gene signatures and use our developed methods to find the drugs. Then finally, we use cell-based to validate our results. So we mining the data from the GEO, gene expression omnibus. Then we perform principal component analysis of the host RNA-seq data. So these three data sets show the conditions related to the interferon beta treatments. And we also calculate the correlation of the host transcriptional response. So the three data sets correlate response of interferon beta treatments. So we use these three data sets to identify the interferon beta induced signatures. So we compare these three data sets. You can see more than 1,000 genes in common. Then we perform the functional enrichment pathway. And here we show the top 10 pathway enrichments of interferon beta signatures. So the first one is interferon signaling. The second one is interferon alpha beta signaling. Then we use our developed systems level method for drug discovery. So the data set have the perturbation chemicals information and also many genes information across different cell lines. So you can see here, this compound activates the genes and the other two compounds inhibit the genes. So we give the score. So we use the data from the connectivity map, which is a genome scale library of cellular signatures that catalogs transcriptional response to chemical, genetic, and disease perturbation. So we use the second generation of CMAP called as LINX. LINX data contain over 1 billion data points. It contains 4 perturbation types, different cell types, such as cell lines or primary cell lines. And also it contains 5 signature types, such as transcriptomics, proteomics data. So here we use the transcriptomic data to find out the potential drugs. So here show the expression-based therapeutic score. For example, DE means the differential expressed gene. So plus means upregulate, minus means downregulate. So REC means if the drug activates the genes, we give the plus. If the minus means this drug inhibits the gene expression. So we give the score like this. Summarize the forward hit, so the same trend. So summarize the forward hit, subtract summarize the reverse hit. So the result shown here, our prediction method show the predict compounds here. These compounds can enhance the interferon beta signatures. And also we found the compounds are associated with interferon beta 1 regulatory recurrence. It means these compounds can upregulate this gene expression. So we compare these 2 data and found these 4 compounds. All of the compounds are protein synthesis inhibitor. So then we construct the drug target network between the interferon beta response genes and the candidate compounds. So from the networks, you can see this compound can upregulate the genes or downregulate the genes. So these 4 compounds upregulate these genes shown here and downregulate the genes. So we also perform the functional enrichment to see whether the compounds can induce the interferon beta signature genes. Then you can see the function involved in the type 1 interferon production and also immune response regulatory signaling pathway. So finally, we validate our prediction using the plaque reduction assay. So we infected the viral E6 cells with the SARS-CoV-2 virus. And after 1 hour, we treated the drugs for 5 days, then quantified our assay results. So the results shown here, the best one is the compound shown in B. This compound has the IC50 below 20 nanomole. So I want to summarize my talk. We developed SARS-CoV-2 drug database to predict the potential drugs with validation information support. And in the second part, we identify the interferon beta induced host signatures and found the repositioning drugs for COVID-19. Then we validate the predict compounds and find the best inhibitory concentration below 20 nanomole. So finally, I want to thank my collaborators and my colleagues, first Professor Chen Qianyu and Professor Han-Chung Huang and Professor Shueh-Fen. And I also want to thank all the engineers in AI labs, also the CEO, Sitwu. And finally, I want to thank my postdoctor, Huang Zhenzong, and my PhD student for the gene expression profile analysis. Thank you.
Thank you, Dr. Wang. Question on the floor? Yes, Dr. Wang.
As a few questions, or all right, I'm going to ask my question. So your analysis is based on interferon beta.
Yes.
And so has it been shown clinically that in fact that should be the target?
So in the beginning, in the infection, initial infection, some clinical trial tests, interferon can rescue and kill the virus. So we didn't did the clinical trial for these drugs, but we will try because very few cases in Taiwan, so it's difficult to test the drugs.
But the initial interferon beta is based on your literature review, your mass pooling of data from the literature. Is that so? Okay,
Yeah.
so that's based on existing literature.
Yeah.
I see. Thank you.
Thank you.
This drug, the 3 candidate drug, you just use this bioinformatics, pick it up. Then what's the next step? You're going to explore
Animal
that
model.
animal model, for example, like a hamster.
Yeah. So we collaborate with Liu Shiren in NHI. Yeah.
Okay.
If we got the budget from most.
Okay. So, any other question? No, that's thank you, Dr. Ram again.
Thank you.